Welcome, everyone, to the FDNA webinar entitled Improving the Molecular Diagnostic Yield Using Artificial Intelligence to Deliver Precision Phenomics. My name is Sarah Riley. I am the Director of Customer Success for labs and bioinformatics companies, and we're glad that so many of you can join us today to hear from two speakers about their experience and outlook on the intersection of molecular testing and phenotyping. Our two speakers will each take the floor for 10 minutes. These presentations will then be followed by a moderated discussion that addresses questions that you, our attendee members, can submit during the presentation. Everyone attending the webinar today is on listen-only mode. You may submit questions by typing them into the Q&A box on the Zoom interface. And when the webinar is over, you're invited to send additional follow-up questions to success@fdna.com. So let's begin. I would like to start by introducing our two speakers, Dekel Gelbman and Kyle Retterer. Dekel Gelbman is the CEO of FDNA, which has turned computer vision technology into an innovative digital health platform and database used by thousands of clinicians, researchers, and labs globally in the evaluation of patients with genetic conditions. Kyle Retterer joins us from GeneDx, where he has been since 2010. He currently oversees the test development and integration, data science, genomic software, and bioinformatics groups. Kyle has authored 25 peer-reviewed articles that primarily focus on whole exome sequencing and the discovery of novel disease genes. His areas of expertise include assay design, bioinformatics, pipeline development, genomic analysis automation, and the application of machine learning to clinical genomics. Kyle, please share your screen and begin your presentation.
Thank you, Sarah. So I wanted to talk today about this concept of precision phenomics and where this is taking us as the sort of scale and scope of what we can do with genomics, this really powerful tool, increases more and more. And so you're probably all familiar with this famous or infamous graph, and it was actually just updated, I think this month, to include the last few years. And you can see the cost of generating a genome is declining dramatically. And we're at this point where you can say, well, we can sequence a genome for $1,000. And the pace of growth has been astounding since 2008. Right? So the age of the $1,000 genome is here, but there's a big asterisk on that, and that's that analysis is not included. It's just generating the data itself will cost you $1,000. But what do you do with all that information once you have it? This is a huge challenge that's still largely unsolved. So exomes, a subset of the genome, so about 1.5% of the genome, is rapidly becoming this routine test. At GeneDx, we've actually sequenced over 75,000 exomes to date since we launched testing in 2011, 2012. We're signing out over 1,000 cases a month at this point. It's our largest growing segment of testing, so more and more testing is shifting towards exomes. People are using it for a wide variety of indications. It's a really powerful tool. And we see, depending on the indication that people are coming in for, we have anywhere from a 25 to a 45% diagnostic rate. If we're talking about our rapid express exomes, which are typically done on NICU patients where we receive a full trio, and they very likely have a Mendelian genetic disorder, and they haven't already had chromosomal microarray or other screening tests done first, we're seeing a 45% diagnostic rate in those cases. Similarly, some other people have published studies or presented work on doing whole genomes in sort of a similar testing scenario, and they're seeing similar positive rates. Most of the extra findings from doing genomes at this point are actually coming from finding CNVs and structural variants, and we actually see a lot of those from whole exome as well. The 25% is sort of if you do, a singleton, you don't have parents to see the segregation data, and maybe it's an older child or even an adult, and they don't have necessarily a Mendelian disease. Based on indication, you may see some variability there. But it's this really powerful tool. Lets you look at basically everything at once. Right? But the big problem with exomes is that the X still means expensive. We're talking in the neighborhood of thousands of dollars per test. And one of the big questions that we face as GeneDx, but I think we face more broadly as a genomics clinical diagnostics community, is now that the cost of data generation has dropped so dramatically, how can we make the power of something like an exome or a whole genome, as that becomes more accessible from the data generation side? How can we make it more accessible to patients who need it? How do we take advantage of all the power we have in this tool and make it available to everybody? There's MRI machines all over the country, right? But we don't just order full body MRIs on everybody all the time for no particular reason. We order focused testing because it's economical. We can't, even though we could, do an MRI or we could do a whole genome on everybody, doesn't mean it's a good idea necessarily at this point. And so showing the inverse of our graph from earlier, we had the cost going down. So at the same time, if you think about how much data we're generating, well, that's been going up tremendously. And you can see a few inflection points here. So around 2008 is when NGS testing really started to take off, and we went from doing single gene testing to doing panels. And the panels got a little bit bigger as we got better and better at this. And then exomes came along, and then at this point we're in sort of the age of the whole genome. That's, I think, still not quite there yet in a lot of ways, but it's coming. And so you'll notice the y-axis here is log scale, and so we've increased from the year 2000 to the year 2015. In 15 years, we've gone up by 1, 2, 3, 4, 5 orders of magnitude, or 6 orders of magnitude if you count whole genome. So we've tremendously increased the amount of data. There's 4 things on this slide. One of them is not scalable, and that's Ben Solomon. He's a pediatric geneticist and the head of GeneDx. It would be great if we had 100,000 more Ben Solomons than we do now to interpret all of this information that's coming out, but we don't. And the reality is that we can't generate that many more people with the clinical expertise and the molecular expertise to interpret this data that quickly. We can get more computers. We can use this magical thing that people talk about called the cloud, right? We can just go and pay Amazon or Microsoft or whoever more money and get more computers. Great. We can go to Illumina and buy more sequencers, or we could even buy a Oxford Nanopore MinION. That's what's up in the top right there. We can generate all the data we want. We can process it, and we can scale those really easily. But we can't scale the clinical interpretation. And on the flip side of that, or even following that, genetic counseling and all these other experiences as well that are important and have a strong human element to them. And so that's one of the challenges we face is, how do we scale things? And one of the answers to that is through focus. And there's 2 ways you can focus. Right? One is by genotype, we're talking about whole exome data, or by phenotype. So genotype-driven focus has sort of become the new normal, but traditionally, actually, it was phenotype-driven focus was the way that things were done, right? So if you thought a patient had Gorlin syndrome, you ordered the correct genes for Gorlin syndrome, and you didn't order an exome and look at everything else. However, a lot of genetic conditions have a very broad differential diagnosis. If you have a 2-year-old presenting with global delays and hypotonia and you think it might be genetic, well, it's a little hard to focus that test. And this is where this concept of precision phenomics comes in. So here's a really simple visualization of this that I sort of stole from someone presented something similar like this at ASHG last month, and I thought it was a nice way to look at this. So the clinical view of this is to look at the phenome, to look at the presentation of the patient and say, this is my search space, and I'm going to look in there for a diagnostic answer. The geneticist view of it might be to just say, well, just show me the whole exome or the whole genome. I'll find the things that are interesting, and then I'll match them up with the phenotype. In reality, the most power happens when you combine these 2 pieces together, right? And that's what you're seeing on the right. So if we overlap genotype and phenotype, then, oh, here our variant becomes red, right? And we see our answer. So we've narrowed our search space. And that's what we're trying to do here. And if we can narrow that search space earlier in the process, so if instead of looking at a whole exome's worth of data, maybe we still generate a whole exome's worth of data because that data generation is not too hard anymore, right? It's the interpretation and analysis that's difficult. But if we take that data and we can focus at the time of ordering, we can improve what we'll call in quotes utilization, right? So what is utilization? It's another word in quotes for it might be value-based medicine. And it's being driven mostly by payers, right? So whether that's insurance companies or hospital administrators or lab directors or whatnot, people care about doing cost-efficient testing. But it's also about utilization of those personnel resources that aren't scalable. One of the big problems we face, though, in trying to do precision phenomics is that people are fuzzy. And what we mean by that is that, well, the phenotypes are fuzzy, right? So two different people with the same disease can present in different ways. And there's, a wide spectrum of sort of analog information, right? It's not digital. And the way we collect that information is also fuzzy. And that kind of fuzziness comes in two different flavors. One is incompleteness, where we maybe don't know everything that we wish we knew. And that's where something like FacetoGene can be a really powerful tool. If you have a patient who's seeing a neurologist and the child has seizures, and maybe they have some subtle dysmorphism, well, the neurologist may not be trained to pick up on that dysmorphism and may recognize that there's something going on there, but may not, fully be able to capture it. And the other is the inaccuracy. And so you can have two different people describing the same thing different ways. And again, this is a place where something like FacetoGene or other metric data can be really powerful. So if you can actually measure, something very simple, right, the circumference of the head, and record it at different points in time, hey, that's much more powerful information than just saying the patient has microcephaly. We can try to reduce that fuzziness, right? So we can do all these great things, but we can never truly eliminate it. And so we also need to invest in this concept of fuzzy analysis, where we allow for probabilities of uncertainty. And so, as I mentioned, there's tools like FacetoGene that can help improve this. Another tool that we've been working on at GeneDx is natural language processing to take the medical records and pull out from that human phenotype ontology, or HPO terms. And so you're seeing that on the right. It's a clinic note, and we've pulled out things, and you can see it's actually picking up abbreviations, and it'll handle misspellings and things like that, and so on, and handle different ways of saying the same thing. And so that's another way to get some of that inconsistency out, and to make things a little more systematic. Now, once we've captured all that phenotype information, if it's in something like HPO that's kind of well-structured, well, we can take that well-structured data and we can do a bunch of fancy math on it and try to model sort of the human approach to it, right? To try to model this gestalt matching of two sets of phenotypes, whether you're matching two patients to each other or a patient to a disease. We can try to do that computationally, and it works pretty well. And so what I'm showing here is a graph of cases where we have a lot of recurrent molecular diagnoses. So these are pathogenic or likely pathogenic variants in genes that we feel fully explain the patient's phenotype. And so there's 4 different genes here: CREB-BP, MECP2, OncRD11, and I2SEC2. And what you're seeing on the x-axis is the rank score of the cases that we have, using that sort of sophisticated computational model that says, oh, there's this much information in this term in the tree based on how often we've seen it in a cohort and how it's associated with the gene and so on. And I won't go into all the details of that. You can see that in some cases, it works really well. So CREB-BP and MECP2 are pretty well characterized Mendelian diseases. And we have lots of cases. So we've got 33 cases of CREB-BP, 67 of MECP2 from exome. And you can see we have a really nice tight distribution, and we're on the left here for the most part. We've got a few, kind of a little bit outliers, but nothing too bad. But then we look at these other two genes, OncRD11 and I2SEC2, it gets much worse. OncRD11, we're still able to put some things up at the top, but we have a pretty long tail. And I2SEC2, we're doing pretty awful, right? We're not even getting into the top half, barely, of known Mendelian disease genes as far as being able to identify patient phenotype to known gene-associated phenotypes. And so we've got a problem here. We've got 32 cases of I2SEC2. We've got 81 cases of OncRD11 where we have pathogenic or likely pathogenic mutations in these genes. And so this is where the artificial intelligence aspect comes in. We know that there's complex patterns in this phenotypic data. We know we have lots of cases to learn from, and we have this data in a well-structured way. So we have the genomic data really well-structured. It's stored in a VCF file, right? We also have all the phenomic data pretty well structured. We've extracted all these HPO terms. We have other pieces of information like age and gender and so on. We even have a little bit of family history, which we're trying to figure out how to incorporate that into our models now. And we can take all of this information because we have so many cases, because we have 81 diagnosed cases of ONCARD11, we can take that and we can use that data as training for a deep learning or neural network artificial intelligence model. The central problem with doing this really well is you need to have good, clean training data, and you need a lot of it. And very fortunately, we do have that, or we're starting to accumulate it. Some genes we don't. Some genes are very, very rare, and some genes aren't well understood at all at this point. But what we're finding is that this works incredibly well for the cases that we have, a decent number of genes, or a decent number of molecular diagnoses observed. And so that IQ-Sec2, the one that was really terrible, I'm showing the same data again in red on this plot, and you can see it's all spread out, centered around 35 or so. And we really want to be up in the 0 to 1% range. We want to be at the very top. We want to say this is the gene. And you can see that's actually happening here from our neural network model. So we took about half of our cases and we trained the model. And we said this is what an IQ-Sec2 patient looks like. It learned which features are important, which ones aren't, and it learned the complex relationships between those. And it's then able to apply that model going forward to say, with an actual probability, how likely it is, given a phenotypic presentation, that this patient will harbor a mutation in IQ-Sec2. And so there's a few ways you can use this. One is that you can use it to do that sort of value-based testing that I talked about earlier, right? Where we can, maybe justify to the payer why we want to order a whole exome, or why we should order a small focus test instead. And so you can see sort of two scenarios here. And I have this sort of cartoony plot on the right here. And you're seeing on the x-axis the number of genes in the log scale again. So 1 gene, 10 genes, 100 genes. And on the y-axis, I'm showing you the cumulative probability that we're going to find the diagnostic gene in that set. And so you can see on the red curve here, to get to 90% diagnostic, we have to go all the way out to all 10,000 genes. But on the blue curve, to get to about 90%, we only need to go 10 genes deep. And so maybe for that blue case, we should just order a 10-gene panel. And we can justify that. And we can say, let's order this test first. We get a 90% chance of finding our diagnostic answer. And then if we don't find anything, maybe it's worthwhile doing that other test instead, right? Doing the full exome analysis. Whereas if you had a case that looked like that red curve, you just had that, 2-year-old child with hypotonia and developmental delay, it probably is a good idea to do an exome in those cases and look at the full picture. And so we can start to use this kind of information as we build these models out to guide testing into the right direction. The other really interesting thing you can do with this is if you have one of these really tight curves, so say you had not 90% probability of being in a list of 10 genes, but a 90% probability of being this one gene, but you didn't find a molecular diagnosis in that gene, well, then maybe it's worthwhile to dig deeper on that high probability gene or on variants of unknown significance in that high probability gene with some functional assay or long read sequencing or RNA transcriptome analysis or some other technique to dig deeper, because you really think the probability is very high that the answer would be there. And so that's, in a nutshell, I think where we could be going with this idea of precision genomics and how we can start to use artificial intelligence and deep learning to leverage all of this really well-structured information that we're starting to collect to improve patient outcomes, diagnoses, and to reduce the cost of testing and improve the value. And with that, I'm going to pass things over to Dekel to talk about FacetoGene and their role in this.
Thank you very much, Kyle. Second time I've heard this presentation, but it's still fascinating as if it was the first time. So let me try to tag along on some of the things that you said. But first, before we go into FDNA's perspective of the future of phenotyping or precision phenomics, I'd like to talk a little bit about AI or artificial intelligence. This is a big word that's been being dropped more frequently in the news and around our circles. And what does that actually mean? So we've heard a number of terms that, from my perspective, are kind of in the same family: AI, artificial intelligence, machine learning, computer vision, deep learning, cognitive computing. And at least from our perspective, artificial intelligence does not represent today cognitive computing in a sense that computers really understand and pose some sort of risk on humanity with the doomsday scenario of rise of the machine. I don't think we're going to see any of that over the next couple of decades at least. But what artificial intelligence is, from our point of view today, is a very good technique to deal with the huge amounts of data that we're seeing. And Kyle showed you how we're aggregating more and more data with the advances in genome sequencing. We're really not coped to deal with that as human beings without applying machine learning techniques. And just by the way, machine learning has been around from the late 50s of the previous century, so it's nothing new. But what we're seeing right now with the increasing power of cloud computing, with the increasing power of mobile devices, and with the increased capability of genome sequencers, is almost a perfect storm for AI to really be applied. So at FDNA, we've started a couple of years ago by applying AI through computer vision, through the ability of computers to look at images and analyze them and detect patterns that humans either can see or it's very challenging to see. Now, geneticists are accustomed to work with pattern recognition because of their unique training over decades of seeing patients' faces and trying to evaluate the human morphology and the recognizable patterns associated with syndromes. But Kyle noted that people are fuzzy. Well, they're not fuzzy, but they're restricted and limited to their own bias. So a clinician would be only as good as the number of patients that they've seen through their lifetime. The machine learning system, the artificial intelligence system, is very similar to that. However, we can create a much larger data set, and the computer can access the data very quickly. So at FDNA, what we've done, and we've deployed that in FacetoGene, we've trained the system on tens of thousands of images, facial images of different syndromes, and our current database has information across 10,000 different disorders. About 2,500 of them have images representing them in the database. And we've trained our system with a neural network to identify those patterns. And those patterns sit in a database so that at any given time, if a clinician uploads a photo, the photo is de-identified. And I'll go into details in the next slide. And those patterns are compared to deliver insights in about 5 seconds. And insights could be highlighting specific syndromes that may be plausible, highlighting phenotypic traits that should be considered by the clinician, and even highlighting specific variants that may be observed in a genome sequence. And here's a more detailed flow of how this works, because one of the main challenges that we as a company have encountered is the privacy. How do you provide this service of facial analysis while preserving patient privacy, the face being one of the most identifiable pieces of information of a patient? So we've broken it into a couple of steps, and AI was extremely helpful here because of the unique structure of layers in neural networks. And in essence, what happens when clinicians use our system is they take a photo with either a mobile device or a camera once the image is uploaded to the cloud, it's automatically secured in a private repository accessible only to that clinician. However, the AI system is able to look at that photo and extract information. It actually turns the photo into a mathematical description that cannot be reverse engineered back into a photo, but has sufficient information to be indicative of the patterns incorporated into that face. All this de-identified information is then stored into a huge database that helps train our system to identify different classes of syndrome. So every time a face is uploaded, it accesses that database. It's able to evaluate the syndromes based on the classifier. And then when a clinician actually confirms a diagnosis, it feeds back to the system and says, you should learn from this sample. And it becomes a system that grows and learns every time a clinician uses it. With this system, we were able to be both HIPAA compliant and EU compliant. And now with the new GDPR rules, we're also ready to meet them when they apply in Europe, and we're ISO certified. So deep learning has really been instrumental for us in this way. So this is how FacetoGene actually works, and I'm assuming that most of you are familiar with the concept. The FacetoGene is both a web application and a mobile application that allows clinicians to upload pertinent information about a patient, including the facial image, and the system provides immediate feedback on the likelihood or similarity of the patterns, we call that the gestalt, of the image compared to the images in the database, as well as other phenotypic traits that are not necessarily tied to the face. And in this example, we're seeing a case of a patient with Soto syndrome. What you'll see is we present cards, syndrome cards that provide information and insights on all the syndromes that our system had indicated a high threshold of similarity. And we have two general pheno scores. One is the Gestalt score, talking about similarity of the face versus the average or mean look of a patient with that syndrome, and the compatibility of the features. We also present information derived from the London medical databases in those syndrome cards, so you can read or evaluate reference photos. And I'm not sure everyone knows, but we've just entered into a partnership with PossumWeb. So PossumWeb will be presented shortly together with London medical databases in FacetoGene exclusively. And we allow clinicians to annotate the features based on our AI. We recommend or we suggest typical phenotypic traits that we infer are most probably observed in that patient, making it much easier for a clinician to annotate the most important phenotypic traits of the patient that they're seeing. And so this is a great tool for evaluating patients for clinical diagnoses. But once you've done that and you've added that syndrome to your list of differential diagnoses, what you really want to do is you want to confirm that with a molecular test. So we've added the capability of sharing the phenotype captured by FacetoGene for that patient with the lab. And I agree with Kyle, the intersection between the phenotype and the genotype is extremely powerful in increasing the diagnostic yield of a patient with genetic syndrome. So with this tool, you can either select the type of test, you can select one of the labs that are partnering with us, or you can invite a new lab if the lab that you're working with is not part of this system, and then the lab gets a notification and is invited to become part of our system. And you can select the lab and send that phenotypic information to the lab, and it's all stripped from PHI unless you actively select to share PHI with the lab. I think this is the point also to mention that FacetoGene is provided free of charge both for the clinicians and the labs. So there's absolutely no fee involved in doing this, and the reason is very simple. We've always been very transparent with that. The more people use FacetoGene, the better our technology becomes, and then we can monetize our technology within the pharma industry and making discoveries, recruiting for clinical trials, etc. So this is kind of a summary slide on and very similar again to what Kyle presented, how we see phenotypic data layered on top of the genomic data, improving the diagnostic yield. We have very similar numbers. These numbers are derived from the PEDIA study, and we've reached very similar numbers to what Kyle has shown. From a subset of, from a cohort of about 1,000 exomes studied in this framework, about 25% of them would have been identified as the causing variant without any phenotypic information. But when you add annotated phenotypic data, as well as our facial analysis technology, you reach above 80% diagnostic yield. So in 80% of the cases, we were able to pinpoint the disease-causing variant as the #1 variant from the VCF, which I think is astonishing. So I think it's safe to say that there is a lot of promise in AI or AI-driven phenotyping and complementing genome sequencing and specifically interpretation. But we're more excited about what the future holds, because what we've shown to date is that with facial analysis, and we all know that facial dysmorphism is a critical component for a subset of diseases. Some would say 2,000, some would say even more. But there's so many more diseases out there, rare diseases, genetic diseases, or even other diseases that are impacted by the genetics. But we've built this system that allows us to take raw biometric data in the form of facial photos and link them, correlate them to the exact variants. Why not do that with other biometric data? And obviously, as Kyle noted, clinical notes have a wealth and depth of data that needs to be captured. HPO terms are standardized, but there are a lot of terms, and so it requires a lot of education and training, and a lot of time is going to pass until clinicians start talking in HPO terms in a standardized way, if at all. So standardizing these techniques through AI is very, very powerful. So a couple of examples of what kind of biometric data we're talking about when we say biometric. So for instance, MRIs. If you replace this image of an MRI with a facial photo, this is what we've done to date. We've connected facial photos with phenotypic traits with genetic variants. Our system is already prepared. It's already ready to process other types of input. So brain MRIs could be indicators for syndromes like Retterer-Windham syndrome, and specifically to those phenotypic traits that are associated with the brain MRIs, like a thick cortex or abnormality in the wide cerebral convolutions. Same thing can go to echocardiograms for CHARGE syndrome, or with X-rays for skeletal dysplasia syndromes. So there's a lot of opportunity here. What we're excited about is that through our network that contains thousands of users from over 2,000 clinical sites around the world and over 130 countries around the world, we've been able to create a community. And this community-driven data aggregation or data curation is extremely powerful. We need to overcome these data silos where data is restricted only to specific geographies or specific communities, and be able to share the data around the world between all clinicians, all labs, all researchers. That's really what we're trying to do here. And with that, I'll give the screen back to Sarah, and thank you for listening to us. I think now is the time for questions.
Thank you. Thank you, Dekel. Thank you, Kyle, for your portions of this webinar. I'd like to move us into the discussion portion of today's webinar. Please continue to submit questions using the Q&A button. We have received a number of questions already, and we will try to get to as many as possible. So let's start out with a question. We have seen joint press releases about FDNA and GeneDx doing work together. Maybe this question goes to you, Dekel. Dekel, can you tell us about this collaboration between the two companies?
Yes, so I think I've touched upon this briefly in my presentation. At some point, we understood that sharing the phenotypic data between the clinic and the lab is critical for the interpretation of exome sequencing and whole genome sequencing. And there's a flaw in the system for communicating that information. But we've realized that within FacetoGene, clinicians already capture a very deep and accurate phenotype file for a patient. And so GeneDx was the very first partner, along with Blueprint Genetics, to work with us on integrating a system where this information can be streamlined from these two different ecosystems to benefit the patient. So we've announced a collaboration where we're going to go for full integration over the next couple of weeks or months with GeneDx. And ideally, at the end of the day, clinicians would start phenotyping the patient with FacetoGene, click a button. This information will automatically go into GeneDx's system, and Kyle can elaborate on the process it will undergo when it hits that system, but eventually it will turn around results faster, cheaper, and more accurately.
Yeah, I'll add a little onto that. So, from the GeneDx side, we've been, phenotyping, especially with regards to our exome cases, all the cases that come in, because the phenotype is so important to the analysis and interpretation of the data. We invest a great amount of effort on the side, but really, we realize that we're reproducing a lot of what's already been done by the clinician, and we're just adding extra layer of that fuzziness that I mentioned earlier. And so, the more directly we can get that information from you, the clinician, the better the information is for us, and the less work it is for everybody involved too. So that's one of the big advantages. The other kind of cool thing I see potentially with this, and it's not something that's going to happen immediately, but is the potential to kind of close that feedback loop also. So in cases where, we may from the lab report back a variant of unknown significance, there may be some follow-up in the clinic on that variant. And, maybe you can go and look more specifically at the patient for, maybe there's a biochemical test, or maybe there's just particular features that you could evaluate, or you'll see the patient, a year later when things have progressed further. You'll be able to tell us, whether or not that variant of unknown significance really does fit the clinical picture or not. And you can close that feedback loop, and then, we can really potentially start to improve things even more.
Great, thank you. We have a number of questions coming in for clarification about, let's see, whether right now the FacetoGene system can accept things such as X-rays, MRIs, and EKGs. And there's questions about whether artificial intelligence would be possible for inherited cardiac disease, for example. Could one of you speak to that?
Well, I'll probably speak to that. So there are a couple of answers to this question. FacetoGene can accept any image file. So assuming this image, the X-rays or MRIs, are converted into readable files that are supported by the system, we can accept them, and they could be logged into the record of that case in FacetoGene. As for the processing, we do not process that yet. We are still in research and development mode. But if the question is from someone who wants to collaborate with us on the research and development of that project, we're happy to do that.
Great, thank you. Let's see. Is it possible to push the output of EHR, LIS, or other systems instead of or in addition to pushing this information to the laboratory?
So I assume this is from FacetoGene. And so the answer is that currently what you can do is you can produce a PDF report of the case, and you can insert that into your other system, your EMR, for instance. But there is no integration yet with any EMR. We're working with some leading EMRs on finalizing integration, and hopefully in the next 2 or 3 months, we'll be able to integrate with Epic at least.
Great. Kyle, this is a question for you. Can you speak more to the perceived utility of genome versus exome testing?
That's a great question. So we are offering whole genome sequencing at GeneDx. We just started offering that this year. We've been, doing it on sort of a research basis for a little while. So far, at least internally, and from all of the work I've seen presented and published, there's pretty minimal additional diagnostic value from a whole genome. Most of what gets picked up by it is, tends to be large structural variants or copy number changes that you could have seen on a chromosome microarray without any real issue. Now, there's still, occasional extra things that you can find on whole genome that you might miss that way. But for the most part, that's where the additional yield is coming from. Every now and then you'll see a deep intronic or regulatory variant that's already well characterized, and you'll get a diagnosis from that. But otherwise, you're mostly adding additional variants of unknown significance. So unless you have a pretty focused search that you want to do, we're not seeing a huge value from whole genome at this point. I think that's eventually going to change, especially as we shift towards longer read technologies for whole genome. There you may see some real value for it. But as long as we're kind of stuck in the short read Illumina world for most of this testing, there's going to be some added value from whole genome, but it's not like it's going to be double the yield. That's not the case.
Kyle, another question for you. HPO is currently the major database of phenotypes, but of course has its limitations. Can you speak to giving us a short summary of the benefits and limitations of HPO, and also whether there are other databases that should be considered?
Yeah, HPO has grown a lot since they first started. They've added a fair bit of complexity to try to address some of the problems. So one of the big initial issues with it was it didn't allow for sort of shades of gray of anything, including progression over time. Now there are modifier terms within HPO that let you tag that data in a structured way, and so you can add an onset to a feature, or you can add actual numerical values even. Now, whether HPO is the best way to do that kind of stuff, you could debate one way or the other. We're finding that there's definitely value in taking the HPO terms as sort of a structured way to describe phenotypic presentation. That's really useful. But that it is, anytime you can actually quantify something, that's even more useful. So if you can, quantify the degree of microcephaly, if you have the actual, QT interval measurements, like that's always better, right, than just saying long QT interval. If you can, put a number to it, especially if we're trying to do machine learning from it. If you have already, introduced all these sort of approximations into the way you're describing things, and then you're trying to infer from those approximations, well, you're going to have sort of fuzzy inference. And so the more precise your information is to begin with, the better, especially as we do these AI techniques.
Great, thank you. Dekel, now we've had several questions come in about consent. Specifically, does a patient need to consent to, in the clinic to upload a picture into FacetoGene? And if not, do you think that there needs to be more consideration for a process that involves consent, such as a signed paper agreement?
So that's really a question that varies from one place to another based on the jurisdiction and the laws that apply. Our perspective and view on this is that patients need to consent to have their photos taken. The consent that a physician needs to collect from a patient, again, from our point of view, is not different when using FacetoGene than what the physician would have taken without using FacetoGene, because in essence, the facial photo, the PHI, remains private for the clinician. Now, within FacetoGene, there are a couple of options of sharing with the community, and that obviously requires different tiers of consent. So if a physician is sharing a case with a colleague, whether within their own team or with remote teams, then if they would have gotten a consent from the patient to do that, they should do that. The same consent should apply within FacetoGene. If the physician wants to upload that case with a photo into the unknown forum, then the consent should reflect that. If a physician wants to contribute that photo into the London medical databases, for instance, then the consent should reflect that. So FacetoGene is a platform, and as such, it does not require a different consent, but the use of this platform is really what dictates the level of consent that the physician should take from the patient or their families. And we do have model consents on our website that reflect any of these use cases.
Thank you. Kyle, this is a question for you. Currently, how are cases pushed to the laboratory? Let me rephrase that. How are the cases that are pushed to the laboratory from FDNA being integrated into the workflow at GeneDx?
So we're still working on some of the details there, but essentially what will happen is right now we have a manual step that takes the submitted clinical records, the checklist, and all the other info that were provided, and abstracts them into our system in terms of HPO terms, along with some semi-structured data. And the idea here is that basically by using FacetoGene, you'll sort of prime that system with all the information that you put into FacetoGene. That'll get passed along to GeneDx, but then we will still do our additional review of that data to ensure that everything is complete and accurate. So it's basically making the existing system better and more complete.
Great. And I think, Kyle, you touched on this idea earlier, but it's an interesting question that I'd like you and Dekel to expand upon. Can you develop scanning tools for the EMR to seek out possible unrecognizable disorders, such as seeking out patients who haven't been seen by a specialist to help with detection for referrals?
I think that's a really exciting idea, and I think we have a lot of the technology that would enable us to do it. It would be finding the right partner willing to do that with their EHR and have the level of consent to do that as well.
Dekel, would you like to comment on that?
I echo that completely. So I think the technologies are here, and we can do that. It all depends on the level of access that we have to the data and the willingness of the EMR as a partner to do this. But I have no doubt that by doing that, and we have, one of our clinical sites and partners, Greenwood Genetics, has allowed us to go and look at all cases. I think that the insights that we can get with today's knowledge, going back even 10 years to existing cases, is just fascinating in terms of the yield of what we learn, and even very beneficial to patients that are still alive and waiting for a diagnosis. So, I think that's where we should go. That's what I have to say.
Great. And to attach to that idea, Dekel, what are some of the recent partnerships for FDNA that highlight the company's future plans?
But I think we've been very, very focused on three stakeholders within the genetics community, the clinicians, and we've been working with individual clinicians and with hospitals to implement FacetoGene as an additional tool in the clinical workflow. We've been working with labs and bioinformatic partners to allow them to access the information and utilize the output of our technology into their system so that they can improve the results. And last but definitely not least, we've been working with a lot of research groups, including patient advocacy groups and research groups within pharma companies to try and leverage our phenotyping technology to make new discoveries, to understand genomic mechanisms and genomic pathways better for specific diseases, try and define new phenotypes for diseases that may have not been characterized by specific facial attributes in the past. Try to even within known diseases stratify different subtle phenotypes and correlate them with different genetic variants. So these are the three areas that we're focused on primarily.
Very interesting. Thank you. Another question that's come in. Historically, genetic tests have been ordered with poor phenotypic information. Has there been significant progress convincing physicians and customers to provide more or complete phenotype information with their orders, because diagnostic labs may provide better interpretation of genomic data?
I think we've seen it.
Yeah, go ahead.
Yeah, at GeneDx, we've definitely seen an evolution of that with our repeat exome customers, where the phenotypic data is really important. So if you just order an exome with no phenotype at all, it's this very unfocused, unguided test, and we'll still of course see known mutations and so on, but some of the interpretation is based on the phenotype. And so people who are repeat customers, we've made a point of messaging to them that this is really important to receive the phenotypic info, but it's also important not just for exomes, but for any test that we're doing. Often if we're interpreting things using the ACMG criteria, some of those criteria are based on phenotypic presentation, and so without that info, we may not be able to fully interpret a variant, and you may be left with more variants of unknown significance than if we did have the phenotypic information.
Mm-hmm.
And I think this is pretty evident from the work that we've been doing with PEDIA that shows the importance of phenotype information to really increase the yield and hone in on identifying those disease-causing genetic variants. We've been hearing this repeatedly from all of our lab partners and bioinformatic partners. In fact, in some cases, some partners claim that the additional information of phenotype is even more powerful than performing a trio exome. So I think that's a theme that we've been hearing for a long time now.
Thank you. We do still have a number of questions left to answer, and I'm very sorry that we are not able to get to all of these. We will try to respond to them afterwards by email. So the last question then, to summarize this presentation, is really about whether there's any final words that Dekel and Kyle you'd like to leave us with about the future of artificial intelligence within the field of genetics.
So I'll go first. This is Dekel. I think artificial intelligence is only as good as the data that it's trained on, and I think that a lesson to be learned from other attempts to utilize artificial intelligence is that sharing data across communities, across geographies, across different stakeholders is crucial in order to get as much data points as possible. We want to get as many ethnicities as possible. And we want to get as many age groups, different genders, different geographies. And only then we would be able to create something that is really standardized and could be utilized by the entire community. So I think data silos are a real risk for the future of AI, but community-driven data is a real benefit and has a lot of potential in the AI world.
Yeah, I'll agree completely with what Dekel's saying, and add that the other part that's really important with AI and being able to make good use of it is that AI is only as good as what we teach it how to do. So it's not just about having lots of data, but it's about having high quality data where we can give it some meaning. And then the AI system is able to learn from the input that we're giving it in a semi-structured way, right? So we need to tell it what we want it to do. We can't just say, here's a bunch of data, do magic. That's not how it works, right? We say, here's a bunch of data and here's what it means. Now you tell us what else you can figure out based on that. That's where the real power is. And it potentially lets us solve that scaling problem that we have now in the world of genomic data.
Wonderful. Thank you, Dekel. Thank you, Kyle, for your interesting and informative perspectives on artificial intelligence within our field today. Again, for those of you who submitted questions that we couldn't get to, you can email them to success@fdna.com, or we will try to send responses in the coming few days. A recording of this webinar will be sent to you in the next few days also, and you can find it on our website when we have completed this. And if you have any questions about today's topic or about FDNA, again, you can email us at success@fdna.com. Have a good rest of your day. Thank you, everyone, for joining us.